clarity clinical solutions.
Imagine running a clinical trial for 2 years. Halfway through, you peek at the data and see the drug looks amazing. Should you stop the trial early and declare victory, or keep going? That is the dilemma of interim analyses. Knowing when to look at the data, and what to do when you see it, is one of the most important decisions in clinical trial design. An interim analysis is a planned look at the trial data before the study is complete. It is not just curiosity, it is a formal, pre-planned statistical evaluation to decide if the trial should stop early, continue as planned, or be modified. The key words are pre-planned. Peaking without a plan is one of the fastest ways to invalidate your results and ruin years of work. The 3 most important aspects of interim analyses are why we do them, how we control the statistical risks, and what decisions they can trigger. Interim analyses protect patient safety, save resources, and can get effective drugs to patients faster. But they also create statistical challenges, specifically, the problem of multiple looks at the data, which increases the chance of false positives. Interim analyses serve 3 main purposes. First, safety monitoring. An independent data safety monitoring board reviews unblind safety data to ensure no patients are being harmed. Second, futility analysis. If the drug is clearly not working, stop early and save patients from ineffective treatment. Third, efficacy analysis. If the drug is overwhelmingly effective, stop early and get it to patients sooner. Each purpose has different statistical rules and different thresholds for action. Every time you look at the data, you increase your chance of a false positive, finding a significant result by random luck. If you do one final analysis, your chance of a false positive is 5%. If you do 5 interim looks with no adjustment, the false positive rate balloons to over 14%. That is why statisticians have developed strict rules, called alpha spending functions, for controlling the overall type 1 error rate across multiple looks. To control false positives, statisticians use stopping boundaries, predefined thresholds that are more stringent than the usual p less than 0.05. The most famous is the O'Brien-Fleming boundary, very strict early on, then gradually relaxes toward the end. At the first interim look, you might need p less than 0.001 to stop. At the final analysis, you use the standard p less than 0.05, but only after adjusting for the earlier looks. Interim analyses are not done by the trial sponsor or the investigators, they are reviewed by an independent data safety monitoring board, or DSMB. This group of experts sees the unblind data, evaluates safety and efficacy, and makes recommendations. The sponsor and investigators remain blinded to interim results. This preserves the integrity of the trial and prevents bias in how remaining patients are treated and evaluated. Futility analysis asks, is there any realistic chance this trial will succeed if we continue? If the answer is no, the DSMB can recommend stopping for futility. A common approach is conditional power, the probability of achieving a positive result at the end, given the data observed so far. If conditional power drops below 10 or 20%, continuing the trial may be unethical. Why expose more patients to a treatment that has little chance of being proven effective? Stopping early for overwhelming efficacy sounds great, save time, save money, help patients sooner. But there are trade-offs. Early stopping means less data on long-term safety, fewer subgroup analyses, and less precise estimates of the treatment effect. Regulators sometimes view early stopped trials with caution. The gold standard is to pre-specify both the stopping rule and the planned number of interim looks in the protocol before the first patient is enrolled. Some interim analyses are used not to stop the trial, but to adjust the sample size. If the observed effect is smaller than expected, you can increase enrollment to maintain statistical power. This is called sample size re-estimation. It must be done with strict rules. Unblind re-estimation requires careful adjustment to preserve the type 1 error rate. Blinded re-estimation, where you look at overall variance without seeing treatment assignments, is simpler but less powerful. Interim analyses have several common pitfalls. First, unplanned looks. A sponsor who peeks at the data out of curiosity has compromised the entire study. Second, too many interim analyses. Each additional look consumes statistical alpha. Third, confusing futility with failure. A failed futility analysis does not mean the drug does not work, it means the current trial is unlikely to show it. Fourth, overinterpreting early efficacy in small subgroups. Interim analyses can use either Bayesian or frequentist methods. Bayesian approaches naturally support continuous monitoring. You update the posterior distribution after each new patient cohort and stop when the posterior probability crosses a threshold. Frequentist approaches use pre-planned looks with alpha spending functions. Both work well when properly specified. The choice depends on the trial design and regulatory preference. First, interim analyses are essential for safety and efficiency. They protect patients and can accelerate drug development. Second, every look at the data consumes statistical alpha. Proper stopping boundaries must be pre-specified to avoid inflated false positive rates. Third, DSMBs are the guardians of interim data. Independent oversight preserves trial integrity and prevents bias from creeping into the results. So, what is an interim analysis? It is a planned peek at the data mid-trial, guided by strict statistical rules and independent oversight. When done right, interim analyses save lives by stopping harmful drugs early, ending futile trials, and accelerating access to effective treatments. When done wrong, they produce misleading results that can harm patients. The difference is preparation, transparency, and discipline.